So before I start, I'd just like to thank the many people involved with the project, in particular my old species supervisor, Richardson, and Juan Illera, who did a lot of the kind of field work and groundwork, the work that we talk about. Okay, so you all know that a lot of the phenotypic differences that we observe across wild populations are the product of differential patterns of natural selection. And increasingly, there are some really nice examples of how natural selection drives differences among populations. So a lot of you will be familiar with the examples from deer mice being adapted to different substrates of the ground, and from sticklebacks being adapted to different parts of the water body. But of course, natural selection doesn't operate in isolation. Also, there are non-adaptive forces going on in any set of populations that we're interested in. And I'm particularly interested in population history. And if we think about how population history might drive differences among natural populations, a great species to consider is our own. So in humans, population history has been mapped out really nicely. And so we know that there have been these founder effects as humans colonized out of Africa, and now these things have been dated really nicely and accurately. And these founder effects have shaped patterns of phenotypic variation even to the present day. So things like variation in skull morphology and variation in disease resistance genes, and therefore our ability to detect and resist infections, they can be predicted with a really high degree of accuracy just from distance to Africa. And these are traits under selection. So what you can do is, once you know how population history affects these traits, you can start to look at how residual variation is affected by natural selection. So those are the questions we're kind of interested in. We don't work on bilingual mammals, we work on little brown birds. And this is an endemic species to the Canary Islands and Madeira. This is Berthelot's pipit. So as I said, it's from this region collectively known as Micronesia, where there are 9 populations here in the Canary Islands, just off of North Africa. These tiny little rocks in the middle of the sea called the Selvagins or Savage Islands. And there are 3 islands up here in the Portuguese archipelago of Madeira. Now, across these islands, if any of you have ever been there, you'll be familiar with this kind of habitat. And Berthelot's pipit is probably the most abundant bird in this particular habitat. And this is a kind of coastal scrub. You see here we've got some PhD students trying to chase one into a net. But there's also an interesting ecological divergence or difference in habitats. So these islands, it's mostly kind of coastal scrub, and then they're volcanic. Up the sides of the mountains, there's laurel forest. And then at the top, you've got these kind of craters, these alpine craters, particularly on some of the Canaries. This is the top of Tenerife. And this plateau is 2,500 meters up. And so you've got populations at sea level and some populations at 2,500 meters where it gets extremely cold. So this species is quite an interesting one to study, divergence and speciation. And one of the reasons that we're interested in it is we're actually looking at the thing, and it behaves more like a wader than a passerine. It runs around and it doesn't fly. And this has been noted since the species was described by this German chap called Woller in 1854, and he noted that it runs rather than flies. And even before that, the locals in the Canaries and Madeira, they gave it names reflecting this. So Corre Camino basically means road runner. And that reflects its preference to just kind of run about rather than fly. It's also interesting because we've already got some divergence that's occurred across the population, so we predict that to be the case because we don't think that it migrates. Indeed, it's classified into 2 subspecies, or it has been until recently. The nominate subspecies, which is in the Canary Islands and the Selvagins, and the Madeiran subspecies. As you can see, they're strikingly different. For a little brown bird, but actually the Madeiran bird's a little bit bigger. And of course, ornithologists like to speculate. But there are more good. So Juan Illera, back in 2006, went out and sampled all of these populations. And he screened them at a mitochondrial gene at cytochrome B. And what he basically found was that they're consistent with a lot of island birds. There was very little variation. Mainly just one haplotype. But there were additional haplotypes. suggested that maybe these birds come over from North Africa into the Canaries, where the subspecies is, and then subsequently colonise northwards. And that it's probably quite recent, given the low levels of mitochondrial divergence. So we took that a bit further with some nuclear data. We first looked at some microsatellites. Similarly to what Juan found, we found that the highest levels of variation were in the Canary Islands, and then you've got these reduced heterozygosity and allelic richness in these northern archipelagos. So that's consistent with his idea of northern colonisation. Wanted to model it a bit more explicitly, and we did that using ABC. And with that, basically, you can plot a whole bunch of alternative colonisation scenarios, test which one's the most likely. And consistent with what we see in the data, we find that the most preferred colonisation scenario is one where there's a northward spread from the Canary Islands to these. We've mapped that in a bit more detail, so we also find that there was probably independent colonisations to all of these northern islands from the south. And then this was very recent, so less than 25,000 years ago, and that less than 50 birds, probably considerably less than 50, that's the upper kind of 95% confidence limit. So it seems that we've had this northern colonisation with kind of independent bottlenecks in the northern archipelagos. So if we look at the effect of that one in terms of genetic structure, this is a PCA of the genetic data. And what we find here, you've got one circle which is 95% confidence limits for each individual island. And what we can see is that the clustering is very much across these archipelagos, and that the archipelagos are all divergent from one another. So then we wanted to take this pattern of genetic structure, look at it in a bit more detail, and see if we can figure out really what's driving genetic structure. Is it just the colonisation history, or is it something else? So in equilibrium populations, if neutral genetic structure is just a pattern of limited gene flow amongst those populations, then we see this famous pattern of isolation by distance, where genetic structure and geographic distance between populations positively correlate. If we look at that in our species, we find that that's not the case. So we thought, okay, well, what about these altitude differences that we find within our islands? So we can actually map that as well. We can see whether the environmental difference between sets of populations, and if there's local adaptation, have we got limited gene flow between those populations and more gene flow between populations that are not adapted? And if we map that, then we find again we get no correlation. Okay, so finally then we want to see whether it is just a pattern of the product of colonisation history of the species. And one way that you can test that is you predict basically, because we've got these independent colonisations in northward bottlenecks, we predict that two populations that have both been more severely bottlenecked are going to be more genetically differentiated from one another than populations that haven't been bottlenecked. So based on that idea, we just took one index of bottlenecking, Garza and Williamson's M ratio is based on the allelic size range. We converted that to a distance metric for pairs of populations and plotted that against genetic structure. And if we map that, we find we get a nice strong correlation between bottleneck distance and genetic structure. So it seems that how bottleneck pairs of populations are, that's just the factor driving genetic differences. So really it's pattern founder effects, at least at these neutral loci. So that's quite nice. We've got our colonisation history and we've got an idea about what's driving genetic structure. And what we then want to do is start mapping either functional genetic or phenotypic traits onto that. So I'm just going to talk about one today. We've done more. I'm going to talk about morphology. So when Juan and when I both went out, we measured all of these birds, the whole suite of traits, from beak length and tail length, and things like that. We didn't kill them. And if we look at some of those data across populations, we do see differences across the archipelago. So this is just some of the raw data. This is Tarsus length. In this particular example, we see that the Canary Islands and the Madeira are inconsistent with our idea of subspecies. So the subspecies is supposed to be this one and this one, and then Madeira and Madeira are different subspecies. We see that in this particular example, the Canaries and Madeira have longer tarsus, and the Savagins have short ones. But But then if we look at different traits, they're not getting consistently bigger or smaller across the different archipelagos. So the bill width, we find that actually it's the Madeiran and the Salvadoran birds with wide bills and the Canaries with the short ones. And then we look at another trait, head length here, then it's actually the subspecies are grouping more similarly together, so the Canaries and the Salvadoran birds with the Madeirans with the longer bills. So both the genetic data and the phenotypic data are actually suggesting that we've either got 3 subspecies or we've got 1, depending on your philosophy. And if we visualize the morphological data in a multivariate framework, so put them all together, the same using a PCA, again we find that our archipelagos group is 3 separate groups. There's a bit more variation within archipelagos than the genetic data, but they are separating out. And what's interesting as well is that if you look at that, we do see a pretty similar pattern of divergence between the morphometric and the genetic data. And sure enough, if you plot patterns of morphological divergence, kind of like a QST across populations, against the bottleneck distance, again you get this nice correlation. You don't get it with geographical distance or with kind of environmental variation across the population. So it seems in this case, at least for the morphological data that we've measured, it's quite a nice example of how founder effects can drive populations apart at the phenotypic and the genetic level. So as I said, that's just kind of a start really of what we're doing, and really what we want to do now is start screening a whole load more genes. We've just had some funding to do some genome-wide variation in these populations, and really start to look at the mechanistic basis of how founder effect and selection might interact to drive populations apart, quantitative and functional traits. So I'll stop there, and thanks for listening. Take some questions.
Are there any plumage differences back when the original subspecies described, or was it just based on size?
No, it was basically just based on the bill. So they just saw that they had a longer beak and didn't seem to measure anything else, and described it on those grounds.
How about just subjectively, can you tell birds apart by looking at them from the different places?
No, it's only really size. I mean, we haven't actually measured that in a great deal, and I think it would be an interesting thing to do, particularly across the ecological gradients. Also, so there is more variation within the archipelagos at the morphological data than the genetic data, and there is some indication that that's driven by habitat differences as well. So the body size variation they do seem to be larger higher up as well. So I think it's a chance to answer that.
Okay. So with your bottleneck influencing FST, it seems like what a bottleneck will do largely will be to bring down variation within populations. And that would increase FST. Does that seem to explain all the sort of bottleneck effect on isolation, or is there anything besides just changing those population level diversity?
Yeah, that's about it, yeah. And then the key is that it's stochastic, because there is independent colonizations. You've got stochastic reductions in genetic diversity in all of these newly colonized populations, and so, yeah, that's it. That's basically what drives divergence. But it seems to be able to drive it at quantitative traits as well, and that's quite interesting. We need to really kind of get into a lot more detail about how that operates. So it's quite long and complicated. I can chat to you about it afterwards, but basically it's based on the Garza and Williamson's M ratio, and just using a simple bit of maths, converting it from a single population